Our universe is filled with wonders, from the vast expanse of space to the intricate workings of the microscopic world. Among these wonders is the molecule of life, DNA. DNA is the blueprint of life. Imagine a vast library filled with books. These books contain the instructions for building and running every living organism on Earth. DNA is like the language those books are written in. DNA is made up of just 4 basic building blocks called nucleotides. These nucleotides are like the letters in the genetic alphabet. They are arranged in a specific sequence, like words in a sentence, to form genes. Genes are like the sentences in the book of life. They carry the instructions for making proteins, the workhorses of the cell. Just like a misplaced letter can change the meaning of a word, changes in the DNA sequence can alter the instructions encoded by genes. These changes are called mutations. By studying DNA, we can learn about our past, present, and future. Mutations can be small, affecting just one single nucleotide in a gene. These are called point mutations. Think of it like a typo in the book of life. Even a single letter change can alter the meaning of a word. There are different types of point mutations, each with its own potential consequences. A silent mutation is like a silent typo. The change in the DNA sequence doesn't actually change the protein that the gene codes for. A missense mutation, however, is a bit more serious. It's like changing one letter in a word and getting a completely different word. This type of mutation changes the amino acid that the DNA codon codes for. This can lead to a change in the shape and function of the protein. The most drastic type of point mutation is a nonsense mutation. This is like changing a letter in a word and turning it into a stop codon. A nonsense mutation can result in a shorter, non-functional protein. For example, sickle cell anemia is caused by a single point mutation in the gene that codes for hemoglobin. Sometimes mutations involve the addition or removal of nucleotides from the DNA sequence. These are called insertions and deletions, respectively. These mutations are like adding or deleting words from a sentence. They can disrupt the reading frame of the gene, leading to what's known as a frameshift mutation. Imagine a sentence where each 3-letter word represents a codon. The cat ate the rat. Now let's delete the first E. The sentence becomes the cat ate the rat. The reading frame has shifted and the sentence no longer makes sense. Similarly, a frameshift mutation alters the way the DNA sequence is read. The protein produced is likely to be non-functional. Our DNA contains repetitive sequences, like stuttering in the genetic code. These are short sequences of DNA that are repeated multiple times. While usually harmless, these repeats can sometimes expand, adding extra copies of the repeat sequence. These are called repeat expansions. Imagine a word in a sentence being repeated multiple times. The cat ate the rat. This repetition can make the sentence awkward but doesn't necessarily make it nonsensical. However, if the repetition continues to expand, the sentence becomes increasingly difficult to understand. Repeat expansions can disrupt gene function. They are associated with several genetic disorders, including Huntington's disease and Fragile X syndrome. Section 5. Chromosomal catastrophes. When things go really wrong. Mutations can occur on a much larger scale, affecting entire chromosomes. These are called chromosomal mutations. Imagine chapters being rearranged, duplicated, or even deleted from the book of life. Chromosomal mutations can involve duplications, deletions, inversions, and translocations. These large-scale mutations can have dramatic effects on an organism's development and function. They can lead to developmental delays, intellectual disabilities, birth defects, and an increased risk of certain cancers. Down syndrome, for example, is caused by an extra copy of chromosome 21. Understanding these mutations helps us grasp the complexity of genetic disorders. It underscores the importance of genetic research and medical advancements. Section 6. Guardians of the genome DNA repair mechanisms. Our DNA is constantly under attack from various sources, both internal and external. These include errors during DNA replication, exposure to UV radiation, and even the byproducts of our own metabolism. Thankfully, our cells have evolved sophisticated mechanisms to repair damage to our DNA. These DNA repair mechanisms are like the editors and proofreaders of the cell. They work tirelessly to correct errors and maintain the integrity of the genetic code. Without these repair mechanisms, mutations would accumulate rapidly, leading to cell death and disease. There are several different types of DNA repair mechanisms, each specializing in a particular type of damage. Understanding these repair mechanisms is crucial for understanding how mutations arise and how they can be prevented or corrected. This knowledge is vital for developing new treatments for genetic disorders. Section 7. Fixing the code directly. Direct and base excision repair. Some types of DNA damage can be repaired directly, without having to remove and replace nucleotides. This is called direct repair. One example is the repair of thymine dimers, which are caused by UV radiation. An enzyme called photolyase can directly reverse this damage by using energy from visible light to break the bond between the thymine bases. Think of it like a molecular undo button, directly reversing the damage caused by UV radiation. Another important repair pathway is base excision repair, BER. BER is like a targeted search and replace function for damaged or incorrect bases in the DNA sequence. It involves a team of enzymes that work together to remove the damaged base and replace it with the correct one. BER is essential for repairing damage caused by a variety of factors, including oxidation, alkylation, and deamination. Section 8. Cutting and pasting for accuracy nucleotide and mismatch repair. Nucleotide excision repair, or NER, is a more complex repair pathway that deals with bulky DNA lesions that distort the DNA helix. These lesions can be caused by a variety of factors including UV radiation, cigarette smoke, and certain chemicals. NER is like a molecular cut and paste mechanism. It involves a complex of proteins that recognize and cut out a short stretch of DNA surrounding the damage. This gap is then filled in by DNA polymerase using the intact complementary strand as a template. Mismatch repair, or MMR, is another important repair pathway that corrects errors that occur during DNA replication. DNA polymerase, the enzyme responsible for copying DNA, is remarkably accurate but not perfect. It can sometimes insert the wrong nucleotide, leading to mismatched base pairs. MMR acts as a proofreading system, detecting and correcting these mismatches. Both NER and MMR are crucial for maintaining the integrity of the genome. Section 9. Double trouble. Repairing double-strand breaks. Double-strand breaks, or DSBs, are the most dangerous type of DNA damage. They involve breaks in both strands of the DNA double helix, which can be caused by ionizing radiation, certain chemicals, and even normal cellular processes. DSBs are particularly dangerous because they can lead to chromosomal rearrangements, deletions, and cell death. To repair these breaks, cells have evolved two main pathways: non-homologous end joining, or NHEJ, and homologous recombination, or HR. NHEJ is a faster but more error-prone pathway that directly joins the broken ends of the DNA molecule. While NHEJ can quickly repair the break, it can sometimes result in the loss or addition of nucleotides at the repair site. HR is a more complex and accurate pathway that uses the intact homologous DNA sequence as a template to guide the repair. HR is more accurate than NHEJ but requires the presence of a homologous DNA template, which is usually only available during certain phases of the cell cycle. Defects in DSB repair pathways can lead to genomic instability and an increased risk of cancer. Section 10. Bypassing the damaged translesion synthesis. Sometimes DNA damage is so severe that it blocks the progression of DNA replication. In these cases, cells can employ a specialized DNA polymerase called Translesion Synthesis, or TLS polymerase, to bypass the damage. TLS polymerases are like specialized repair crews that can navigate through difficult terrain. They can incorporate nucleotides opposite damaged bases, allowing DNA replication to continue. However, TLS polymerases are more error-prone than replicative polymerases, and their activity can introduce mutations. Think of it like trying to copy a damaged blueprint. You might be able to make out some of the details and continue drawing, but you're more likely to make mistakes. TLS is a double-edged sword. Understanding the regulation and activity of TLS polymerases is crucial for developing strategies to prevent and treat cancer. Section 11. The importance of fidelity, mutations, repair, and the future. Mutations are the driving force of evolution. They introduce genetic variation, providing the raw material for natural selection to act upon. However, mutations can also be detrimental, leading to disease and even death. The balance between mutation and repair is crucial for life. Too many mutations and the genome becomes unstable, leading to cell death and disease. Too few mutations and evolution grinds to a halt, leaving organisms vulnerable to changing environments. Understanding DNA mutations and repair mechanisms is fundamental to many areas of biology and medicine. This knowledge is crucial for developing new diagnostic tools, therapies, and preventative measures. It's about unraveling the secrets of our genetic code and harnessing that knowledge for the betterment of humanity.